But I wanted to say welcome on behalf of Great Pond Foundation, Chilmark Pond Foundation, and the Island Boards of Health, who've been working collaboratively on and off since last September to try to figure out how do we wrap our heads around cyanobacteria monitoring. The plan for today is to have a presentation from various members of this group, MV Cyano. There's going to be a presentation at the beginning that a bunch of us are going to speak during. And if you have questions during the presentation, please feel free to put them in chat. And Julie, can you wave Julie or say hi? Julie will be taking all the questions and sort of making a master list of the questions that come up. We'll focus on those questions first after the presentation, and then we can, in the remaining time, take any questions live that are left. I just wanted to say first a thank you to all the members of the Boards of Health who, at a time when they're dealing with COVID on top of their regular job, have found a way to make this new program for our island. Because oftentimes the job of Boards of Health is dealing what is there, whether you have time or not. And cyanobacteria and climate change on top of a global pandemic is an awful lot to deal with. So I just wanted to say thank you for your time and your efforts to bring our island together. This is incredibly helpful. I wanted to also thank the Chilmark Pond community for participating, for being here, for speaking up. Chilmark Pond Foundation for being such incredible partners, helping Great Pond Foundation to bring this program together. And I wanted to thank members of my team, Julie Pringle, David Bauk, and Becca Eyrich, as well as Great Pond Foundation's leadership, because they let us focus on this really important task of figuring out how to understand cyanobacteria. And they invested in a FloraProbe, a new technology that let us get data that we've never been able to on island. So thank you to Great Pond Foundation's leadership. So to get started, let's see some pictures and some data and introduce our team. So I'm going to start by sharing my screen. And I was remiss, I didn't introduce myself. My name's Emily Reddington. I'm the Executive Director of Great Pond Foundation. I'm a biologist and I'm an Edgartown resident. And I care about cyanobacteria, not just because it's interesting and we need to know about it, because I swim in the water and my child swims in the water and my dog swims in the water. So knowing what these organisms mean, how we can interact with them and how we can be safe and still use our precious bodies of water is really important. And just simple terminology, cyanobacteria are also known as blue-green algae. They fit into a sort of heading of harmful algal blooms, although they're not algae. They're organisms that when they grow rapidly or bloom can have impacts either on the environment or people's health. In this case, we're worried about people and animals' health. So MV Cyano, we're going to talk about today what it is, and we're going to answer a bunch of questions. What is it? Who are the partners? Why does this exist? Sort of the challenge and the solution. Where and when does monitoring take place? How does the monitoring system and messaging system work? And then at the end, questions and answers. And to get started, Omar Johnson from the Board of Health in West Tisbury will explain to us what MV Cyano is.
Hello, everyone. I'm very excited to be here today, very excited about this program. And, first of all, I just want to say that, this is just a great collaborative. And I'm just super excited, that we're able to have this right now. But what is MV Cyano? First of all, it's a creation by the Great Pond Foundation. And it should be noted, it is the first cyanobacteria monitoring program on Martha's Vineyard. So we should be very proud of that. MV Cyano is a collaborative initiative to monitor cyanobacteria on Chilmark, Tisbury, and Edgartown Great Ponds. And for those who may not know, cyanobacteria, also known as blue-green algae, is a naturally occurring phytoplankton for millions of years. It's been around forever. It's important to know that these blooms or rapid growth of cyanobacteria can have concentrated cyanotoxins. They're triggered by heat and nutrient pollution, and they've occurred in Chilmark Pond in 2018 through 2020. So we have had some issues with cyano in the recent past. MV Cyano will be using color-coded systems of alerts and will provide up-to-date risk information and recommendations. Emily?
Thank you, Omar. And Marina Lent of Chilmark's Board of Health will now take us through who the partners are and their roles.
Okay, so as you can see, and I just want to say how grateful I am that this exists. And what I say about our respective roles will make it clear why. What we have here are boards of health, and you've just met Omar. Matt Poole of the Edgartown Health Department is unfortunately unable to be here today, but has been probably among the leaders in public health on this issue. And myself, I work for the Chilmark Board of Health. We are so glad to be able to work together with the scientists of the Chilmark Pond Foundation and the Great Pond Foundation. This is one of those areas where science and the government decision makers should be working hand in hand in an ongoing partnership, because we have clearly delineated responsibilities. The role of the scientist is to find out exactly what is going on and why it's happening. Their role is not to decide what society should do about it and to bring that about. That's our role in government. So, but without the scientists, we're guessing. I mean, 4 years ago, I was sending frantic photos to Sherry Casso at the Vineyard Commission, Martha's.
We're back in action.
Okay. So I had gotten to the point of saying that as government officials, we rely on you for good information, and it enables us to make good decisions about what needs to be done to keep people safe. And so that was about 3 slides in.
Can you not see my slides?
We can, oh yes, there we go.
Okay. So thank you all for your patience. Sorry about the technical glitch. That's rather embarrassing. What we're back to in our question list is, why does MV Cyano exist? And we're going to start with the challenge. What are we dealing with? Why does it exist? And I'm going to turn that over to Chilmark Pond Association President Amy Salzman. Amy?
Thanks, Emily. Thanks a lot. I just want to comment on the collaboration that Marina was just talking about, because when you lost power, she was ready to just jump in. She said, I've got the PowerPoint, we can make this happen. So the collaboration was just live for all of us to enjoy. But on to the bad stuff. I was tasked with the why we need to do this part, which is the dark side of cyanobacteria. And so first of all, I am the current, as Emily said, I'm the current president of the Chilmark Pond Association, which is the riparian owners group around Chilmark Pond. And our job is to, quote unquote, properly drain the pond, which is a practice we learned from centuries of successful fisheries management from the Wampanoag people, and one Chilmarkers have been using since the 1600s, a long time ago, both to manage the fishery and to protect the surrounding meadows, marshes, and homes from flooding. As a community, we love Chilmark Pond. We care deeply about it, and we have been distressed, is not really quite strong enough a word, as we sort of have sat back and watched its health deteriorate. The reason its health is declining is primarily because of coliform bacteria, basically poop, that has in fact led to the closure of the Lucy Vincent part of the pond where my kids learned to swim, but it's been closed for years, so other kids aren't able to do that. It has also led to closing of the entire pond for fishing and shellfishing for decades by the state. It's also because of excessive nutrients, as Omar suggested. By this, I mean, we've got too much nitrogen and phosphorus, which come from a variety of sources, from septics, it's the P side on the septic side, to fertilizers and animals, acid rain, all can deposit too many nutrients into our pond. Plus, as we all know, it's been warmer and drier as the new normal. Some people attribute it to climate change. I'm one of them, but some don't yet, which encourage rapid growth by plants like cyanobacteria. And as they degrade and turn into muck, those plants use up a lot of the oxygen that's in the pond naturally that other critters need to survive. The other problem is Chilmark Pond is small and shallow, but our watershed is enormous. It goes all the way to the North Road at Menemsha Crossroads and comes down. And then there's nature. Nature, as we all know, wants to close the pond. She's pushing hard from the ocean side to move the beach to the land. So it's a struggle to keep our pond healthy. The other thing I wanted to say is, while Omar did actually a great job describing cyanobacteria, I just wanted to add one thing, is that it was the creature, I guess it's not a creature, it's a phytoplankton, that invented photosynthesis. So we wouldn't be alive on this planet without cyanobacteria. So it's not all bad, except when the conditions that we have now laid for it as humans have exacerbated and accelerated its growth. So as Omar said, for the last 3 years, or Emily said, we have had in Chilmark Pond toxic cyanobacteria blooms. And last year, someone in our community experienced very significant health effects that are consistent with a toxic cyanobacteria exposure. So I get, needless to say, especially after last summer, lots of requests from folks who want to swim in the pond, who want their dogs to go in the pond, who want to boat in the pond. Is it safe? Is it safe? Is it safe? And I'm just thrilled and delighted to know that now we will at least know whether it's safe because of this collaboration, MV Cyano. So I want to thank, on behalf of all of us Chilmarkians or Chilmarkers or whatever we call ourselves, I want to thank first and foremost our boards of health. Marina, I don't know how you do it. And Omar, back at you. It's just crazy. Emily and her team at the Great Pond Foundation, who have taken the ball and run with it, the entire town of Chilmark and all the members of the Pond Association, the MV Commission that was our go-to in the early days on everything, and they've just been fabulous. The Ables Hill Association, who has a water group that's been testing and monitoring and sharing information with us and pushing us forward to do more. And I have to also thank the Chilmark Pond Foundation, which grew out of the Foundation Association, brother and sister. And they just decided to take action and put their money where our mouths have always been about doing something to improve the ecological health of the pond based on science. But we're not done. And the Pond Foundation is going to keep doing this with Emily and her team, because we need now to figure it. Now that we're monitoring what's going on in the pond, we need to start doing stuff to stop it or to improve itself. So I think now it's time for Alan, right?
Right.
Okay, thank you, everybody. And really, this is fabulous.
Thank you, Amy. Again, I'm Alan Holt. I am the current president of the Chilmark Pond Foundation. Been coming to Chilmark for many, many years. We own a home here. We are fortunate to live on the pond and have enjoyed it for so many years. However, as we all have, we have seen and experienced a decline in the pond's health for the many reasons as Amy just discussed. And I remember back, I don't know, it was 2016, 2017, meeting with other Chilmark Pond residents about our mutual concerns related to the pond. We even at that time asked Emily Reddington, who at the time was the science director of the Edgartown Great Pond, to meet with us. And the reason we went to Emily is because what we were looking to accomplish, or what we thought we were looking to accomplish, was already achieved at the Edgartown Great Pond. And there was really no need to reinvent the wheel. We already had a great example of what can be done and what was done. And a lot of these discussions led to the 2018 incorporation of the Chilmark Pond Foundation as a separate 501c3. That was the easy part. Now comes the hard part. We came up with a mission statement, and it is, a community-centric organization which is established to support the science-based restoration of the Chilmark Pond's ecological health and really to protect it for future generations. We also realize that this is a project and that this of great magnitude, and the restoration could take 5 to 10 years of actions to really achieve the goals we are all seeking, which is a healthy pond, which has fish and crabs and oysters. So that is what we're striving for. But even though it's a long-term project, the restoration is really a combination of some very short-term goals, which are focused on immediate improvements to the pond's ecosystem's health. Some of that, an example of that is dredging to improve circulation and flushing during the openings of the pond, and longer-term goals that will identify and ultimately address the causes of impairment within the watershed. We believe it's an effort of a community partnership that we can begin to see immediate improvements. So I can't stress it enough. We all have a role to play in this. And look, generations of families have made some of their most cherished memories on the shores of the Chilmark Pond. And restoration of this community treasure is our legacy. And we need all the support we can get. We have a lot of money to raise to accomplish this, but what I'm most excited about and pleased with is the partnership across the many towns of Chilmark. That this isn't just a Chilmark Pond project. This is a project that will incorporate the efforts of lots of people. The partnership we've already signed up with the Great Pond, Edgartown Great Pond Foundation. We've already seen the benefits of that, and so we hope to continue on this path. Emily, I'll turn it over to you.
Thank you so much, Alan. So partnership's really important because our ponds don't end where town lines are. Our watersheds cross lots of boundaries, and some of our issues are shared. And some people have asked about how does Great Pond Foundation versus Chilmark Pond Foundation work? I feel like I'm talking about from a comic book, but it's like with our powers combined, we can do more. We have greater capacity if we can bring our scientific teams together. And so that's what we've done. We're working with shared scientific teams to tackle shared ecological challenges. So what I want to do right now is introduce Chilmark Pond Foundation's really gorgeous new website. So you can check it out and see some of the things that Alan was talking about. If you go, not just me, but if we all go to chilmarkpondfoundation.org, you can see this incredible website and beautiful drone footage by Tim DeWitt. What I want to highlight are two things. If you scroll down, you can join the mailing list. What this will do is give you updates about the state of cyanobacteria in Chilmark Pond, and it will also give you other news about what's happening around the community. If you go to our pond and you scroll down, you can see the up-to-date maps, which I'll explain where the information came from and the data behind it. if you go to our work, you can learn about the programs that Alan mentioned, which are focused on restoration, community, and legacy, including the Cyanobacteria monitoring program, hopes of establishing a dredging program that's currently under review, and an ecosystem monitoring program. And there's Julie on Chilmark Pond. So let me take us back to the slides and get into more of the nitty gritty about MV Cyano and how it works. So back to the questions. When and where does monitoring take place? What ponds, seasons, frequency, depths? First, to get us started, we're looking at surface samples for cyanobacteria. So in terms of depth, we're looking at the top because many of these, what they call them, scum-forming cyanobacteria are on the surface. We're looking at this year Chilmark Pond, Tisbury Great Pond, and Eggertown Great Pond, including Cracktuxet Cove, which is sort of just on the other side of Eggertown Great Pond Sluice Way. And if you've just entered, perhaps adding mute might be good. All right, so let's get into the specifics. Looking at Cracktuxet, we sample on these 5 stations, and then for reference, the southeast corner of Eggertown Great Pond is here in the sluice way. We also look at all of Eggertown Great Pond at all these stations in the blue and green dots. We're out there on a weekly basis, often twice a week. We also are working with our partners at Tisbury Great Pond and sample all these stations. And one thing I want to point out is different parts of the pond represent slightly different ecosystems. Some are fresher because of rivers and streams coming into them. Some are saltier because they're closer to the breach when it's open. And so it's important to not just sample in one location, but see what's happening all the way around the pond. And at Chilmark Pond, we're sampling in all these stations in the middle pond and the lower pond, and with less frequency in the upper pond. But we're here a minimum of 1 time a week. So when do blooms occur? What season? And what happens is blooms tend to occur when it's hotter in water that's fresher. It used to be thought that the water had to be less than 10 parts per thousand salinity. The ocean's about 32 parts per thousand, but folks are seeing them now, blooms occurring even in salinities of 15 parts per thousand. The more nutrient pollution is present, sort of this more intense the blooms could be because they're like fertilizer for an algal bloom. And if the water's stagnant, that can also be a condition where blooms could form. We typically think we're going to see them May through October, but that depends on how much pollution's in the system and how warm the system gets when. In Chilmark, we've been seeing them in later summer. Okay, now how does the program work? So the cyanobacteria monitoring program we're doing is a tiered approach based off of Dr. Gobler's work in Long Island. We're focusing on fluorometry, which means putting colored light into a sample from a pond that lets us know what plankton are present, including how much cyanobacteria, the concentration in the cell. We're looking under microscopes, and we're also looking at the environmental conditions, the nutrients, the temperature, the light, the salinity. Today, we're going to look at data from the fluorometer and from environmental conditions. And now into the pictures. This is Sam. He was an intern for our program for 3 years. He's holding what we call a CTD, a conductivity temperature depth probe, and he puts that into the water. It can look at the salinity, the temperature, the pH, the water clarity. And so this takes measurements in the water on site. And this over here is the fluoroprobe that Great Pond Foundation purchased. And what it does is you take a certain volume of water, put it into a very clear glass cuvette, and light waves pass through it. And it lets you know how much chlorophyll is present and the concentration of cyanobacteria and other types of plankton cells. Why this is important is we can track not only their abundance at a specific time, but if that abundance is increasing at a rapid rate or blooming. So before you get overwhelmed, these are the data that we look at. I'm going to break it down, but this as a scientist is what we get all excited about. First, at the top with this blue bracket, these are the data we get from the fluoroprobe. They tell us the concentration and micrograms per liter or also in cells per milliliter. Then we look at the probe data, which tells us temperature and salinity. We also use GPS, so we have exact coordinates where we are, and we put all these information together they feed into making that color you see on a map for MV Cyano. So where do those colors come from? This is a little bit overwhelming, but this is a matrix that the Boards of Health worked with Great Pond Foundation to basically take World Health Organization, EPA, and Massachusetts regulations and put the science and the data into risk levels and messages for the community. You don't ever have to see this figure again, but I want to let you know that's where the colors are coming from. So if we look at Chilmark Pond and CHP 01 through 7 and upper, these are the stations that correspond to the same stations used by the Martha's Vineyard Commission and in the MEP. What I want to highlight are the concentrations of blue-green algae cells. When we see 5 and above, we code that as a yellow, which basically that's the next step. The green means no bloom forming. Yellow, orange, and red. Yellow just basically means we're sort of in bloom season. This is sort of your alert that something's happening. We're also looking at the total chlorophyll. That's how much green pigment is in the sample. That can be from blue-green algae, but it can also be from green algae. One of the characteristics of our matrix and one of the standards for looking at blooms is anything above 10 micrograms per liter is considered higher than ideal and you should pay attention to. So we've highlighted those here. And then the number of cells, we're in the 5,000s here. The salinity, anything above 15 we highlight as yellow. And so what that translates into is most of our stations are highlighted in yellow and some are, there's no indication of a bloom. And so what you will see is this map. And right here, all these yellow stations correspond to a cyanobacterial alert. And that basically means it's bloom season. It could occur. We don't have a bloom taking place, but it's possible right now because the conditions are make it so. If we go down to crack Tuxet, we can look, there are even higher levels of blue-green algal cells. The total chlorophyll is above that 10 limit, so it's higher than ideal. You have even greater counts, and the salinity, all but one station is above 15. So that's sort of that critical limit we talked about. So if we were to code those stations and we send all these data and all this information over to the Boards of Health, and then the Boards of Health interpret if these data do in fact correspond to these tiers. What we've created is a proactive versus a reactive system where we're looking at how things change in time. Because in the past, the advice was if the water is green, stay out of it. What we're trying to figure out is what it looks like before a bloom's occurring and getting an indication if a bloom's on the way. If you go to Edgartown Great Pond, the concentration of blue-green algal cells is 0. So we've coded that green, no bloom. There's one station where the total chlorophyll is above what we'd like to see. Of 10, it's at 13. And that looks like it's due to some diatoms here. Those are the only thing with the highest concentration. And the salinity, all but one station is above 15. So what that does is gives you green dots around the pond. If you go to Tisbury Great Pond, you don't have blue-green algal cells, but what you do is have high levels of chlorophyll. So that's indicating that something's making another type of plankton bloom or grow in large numbers, especially at this station, Tisbury Great Pond 3A. But if you look in the green algal cells, that's where a lot of the organisms and the diatoms are coming from. They're not blue-green algae. So what this data lets us see is what is in the water, not just that there is something there. And that's really informative. And so for blue-green algae, all these dots are green. And if you're wondering where 3A is that had that high amount of chlorophyll, that's up here. So how do these data sort of relate to the real world? What we need to pay attention to is the messaging. So green means bloom not present. The yellow is cyanobacteria alert. Basically, it's the season where blooms are possible, but swimming's okay, boating's okay, paddling's okay, fishing's okay, wading's okay. But there's a risk to pets, humans, and livestock ingesting the water. So once you're in bloom season, it's good to not ingest the water and to keep your pets away from it and livestock. And this whole sort of messaging approach, this is from the Boards of Health, how you take the data and turn it into what's safe and what's not safe. And they invested a lot of time and energy to get clear messaging. So if the numbers increase in terms of concentration of cyanobacteria and the conditions worsen, then a bloom watch could occur where boating's still okay, but using caution for some activities and advising against others. And as you go up into the advisory, basically boating is the only thing that's considered safe. So if we were looking, this is the current map for Chilmark Pond based on the most recent data. If you look here, there's always the date that the data were collected. What you'll see on the pond shore is a sign that looks like this, that'll have sort of images of what's okay, what to use caution for, and what's advised against. So what would happen is Great Pond Foundation would put the data together, send it to the boards of health. The boards of health would make a judgment call about what messaging goes out to the community. And then on coordinated websites, the boards of health, Great Pond Foundation, Chilmark Pond Foundation, the Chilmark Pond Association, and now WMVY will all have these color-coded maps present. So looking more specifically, an alert sign would look like this. A cyanobacteria bloom watch sign would look like this. And a bloom advisory would look like this. So to get more information, you can go to greatpondfoundation.org /MV Cyano, and you'll be able to see the same chart, our partners, and the current maps. And the same data will be on all our partners' websites eventually. At this point, we'd like to follow up with some questions. Julie Pringle, do you have any questions that have been coming up for a lot of people?
There have been no questions in the chat,
Okay.
as far as I can tell. So I think if anyone has a question, feel free to fire it away.
Julie, I just have one comment. I haven't introduced a few of my partners in this venture, and that we met many years ago starting this, and that was Ellen Jewett, Doug Sachs, and Amy, even though she was in her role as president of the Chilmark Pond Association, has agreed to join us. So it's really been the four of us who have been working together, and the three of them do much more work than I do, though. That's all I have.
Does anyone else have any questions?
I have a question.
Hey, Dudley, what's your question?
You mentioned cyanobacteria appearing in saltier areas of the pond. Do those cyano possess the same toxicity as those that grow in the fresher water?
So there's cyanobacteria in fresh and in marine systems and in brackish systems. There are even cyanobacteria on dry land. The species that tend to bloom and produce cyanotoxins tend to be in fresher water. So there are marine species, but those don't tend to bloom or create the cyanotoxins. So we're talking about species that can bloom in both fresh and brackish water. And the Vineyard's unusual because most places are not monitoring cyanobacteria blooms in brackish systems or systems with fluctuating salinity. So it's something the state of Massachusetts hasn't quite wrapped their head around, which is why we've modeled a lot of it after the Hamptons, which are dealing with ponds with brackish water and fluctuating salinity. And so it's different, often different species, but there are cyanobacteria in all those ranges of salinity. And the bacteria that exist kind of change depending on the environmental conditions. The populations can fluctuate.
I have another question. I was wondering if there are some kinds of algae that will inhibit the growth of cyanobacteria if they're in the same pond. Will they outcompete them? Will they, exude chemicals that inhibit the cyanobacterial growth?
That's a really good cutting edge science question and one we're interested in. So we turned to microbial ecologists to answer that type of question. And our colleague, Dr. Caroline Fortunato, will be out this summer taking genetic samples from the various ponds to see before, during, and after blooms how the populations change. Because that's sort of evolving in the field of scientific literature right now. when different community members are present, how much cyanotoxins are produced, and how the communities kind of change. And so you're asking a question that scientists haven't answered yet, Dudley, but it's a really good question.
Thank you.
And Jonathan has a question. You'll have to unmute.
Hi, thank you, Emily. This is just a great presentation, and I appreciate everyone's work to get us where we are with the monitoring program. Really, it's fantastic. I have a question to ask Marina in terms of the color coding. Let's just say, as an example, when we code yellow, here's my question. Is it only the ingesting of water that poses risk?
So maybe we should,
Yeah.
we can either pull up the yellow sign or I can just go through it. The yellow sign indicates that enough of the parameters would allow for algae to bloom, so that we want to let you know that it's possible. Because again, the map that you will get with the yellow dots and the green dots and the orange dots is only from the moment that sample was taken. And those blooms can move fast. They can move in different locations apparently, and they can intensify quickly. And 3 days later, it'll be a whole different story. So this yellow alert only tells you that we saw enough parameters for us to say, you know what? If a bloom wanted to take off, it probably could. It does not mean that a bloom is present at this time. So the water is still basically fine. That's why there's check marks on the guy standing in the water, the check mark on the guy swimming around. But ingestion is always a risk in a sense in blue-green season. So we just want to make sure. I would never let my dog drink the pond water in anytime after May, you know, after it gets warm, just because. And so we've put those, and drinking, and, you know, we've put those as just simple prudent precautions, not as an indication that you will become intoxicated from drinking the water. Did I get it right, Emily?
Absolutely. And if the orange tier actually corresponds to drinking water limits. And so yellow is before you've gotten to the drinking water limits. Red is recreational swimming limits. And so like Marina said, a bloom's possible. So staying out of the water, not drinking the water and having your pets away during bloom season is the most cautious and appropriate thing. But measurable at a certain level for drinking water would be orange.
So we're being very conservative. If these are drinking water limits that they've set at orange, we are being conservative. But because we're only taking a point in time measurement, this is not continuous, this is not daily. Therefore, we want to be conservative because we're talking about dogs and kids. And who doesn't love dogs and kids? They're the ones who are going to drink it. You know, I wouldn't, you know, they're the ones who are likely to ingest water and livestock.
Thank you. Thank you for clarifying. Thank you, Marina.
And we just
I have a
we got a question through chat from Neil. He was asking how our monitoring program, MV Cyano, is similar or different from APC and other agencies in Massachusetts. And so I alluded to that earlier. We attended the Department of Public Health and other talks given by Massachusetts and asked for advice for our very strange brackish ponds with shifting salinity. And some of the questions we asked are not normal because most of the monitoring is set up for freshwater systems. And so Dudley alluded to the fact that organisms, phytoplankton, can change depending on the salinity. We are basing our program off of ponds in the Hamptons that have brackish, or they have the salt influence. And basically what organisms can live depend on how much salt is in the water and what the temperature is. And so it's a little bit different because it's not just fresh water, it's freshwater, salt water, and brackish water. And the fluoroprobe lets us look at the concentration of cells in all those types of ecosystems. And someone else had a question. Chris, you'll have to unmute.
Thank you. Yeah. Emily, I have two things. One is a comment and one is a question. The comment is about, I think it's called Upper Chilmark Pond, the third little pond up right behind Lucy Vincent Beach. If you're looking at the cyanobacteria blooms that have taken place at the mouth of Doctor's Creek, where it enters into the lower pond, most of the fresh water is coming from the upper ponds, the two brooks that lead in. And right now you're showing a testing site. I know the MVC also does a testing site in the middle pond, but nobody's doing the upper pond except for the Board of Health, and they're not doing cyanobacteria testing at that site. At least I don't think they are.
So that's a great pond, Chris. Great question, Chris. And sorry, I'll turn it over to Marina.
Well, wait, I don't want to leave it. That's just a comment. I want, my hope is you'll get there.
We're going to be there a few times this year. That's all I was going to say. We don't have the time or staff to be there every week, but we will be there a few times this year so we can get some baseline data.
Good. That's important, because the more data that we can parse, the better. The question I have for you is for your fluoro probe. How easy is it? How costly is it for you in time and money? If people bring you samples, can you test them right there?
We can absolutely test them. The device itself is expensive, but the reason we invested in it is because the information we get from it is really useful. It's the same equipment that the EPA uses. And so if you wanted to bring us a water sample, Julie could provide the right type of bottle and instructions. Omar actually is going to drop off some samples on Monday, and we can process them that same day for $100 each sample. So we are able to get the data within a minute or two. It's pretty fast, but the equipment itself is expensive, and the expertise to run it is a little bit complicated. So basically it's a resource for the island, and we're really grateful that Great Pond Foundation's board was willing to invest in this, because it lets us run as many samples as we want really quickly.
But you're going to charge us $100 a sample. Is that what you just said?
For anything beyond the systems that we're currently working on, yes.
Okay, thank you.
You're welcome. Sarah Glazer had a question.
Yes, I just wanted to clarify something you said about drinking water limits at the orange level. Are you saying at the orange level, it would exceed the safe water drinking limit that's set by the EPA? Is that what you're saying?
Mm-hmm. Yep, exactly.
Thank
Marina,
you.
did you have more thoughts on that? Okay, and in the chat, Elizabeth asked, during the yellow alert, is it safe to consume fish and shellfish in the pond? Let me bring that back up. And we'll be able to post these signage online. So during the yellow alert, everything is safe except for ingesting the water or having your dogs in the water, and they tend to lick their paws and things. So just them walking in the water can be the equivalent of ingesting. At least that's what we heard from our dog officer colleague, Dr. Buck. How frequently will each pond status be updated? This is a question from Chris. At first, I think it's a weekly basis. If we start to go into the orange and red tiers, we're going to try to update twice a week. And the process is the data gets compiled midday to early afternoon, sent to the boards of health. The boards of health review it and then issue the joint sort of messaging that we can all post on our websites. So it's about a 24-hour turnaround. Marina, was that wrong?
Sorry, I'm talking to my dog. She's asking for a walk. Saying no.
I was all worried.
I have a question.
Go for it, Greg.
Is there a level of cyanobacteria above which it's not safe even to boat?
So do you want to talk about the black level, Marina, or do you want me to go over that?
You go over it.
Okay, so we were looking at models from systems that were warmer and even dirtier than the Vineyard. And in New Jersey, they have a black level. And that's where you wouldn't go in the water at all. You wouldn't want your propeller stirring things up. You wouldn't want to go near it. And so what we don't know is how concentrated our cyanobacteria and our cyanotoxins get during blooms on Martha's Vineyard. Because it's cooler here and not at the watersheds aren't as developed as places like New Jersey. We don't expect that to be an issue, but we won't know for sure until we actually have data where we start not only measuring the concentration of cyanobacteria, but measuring the cyanotoxins. And as part of this protocol, we'll be sending samples both to Dr. Gobler's lab for cyanotoxin analysis in Long Island, and we'll also send some samples to the EPA, as many as they're willing to take. And that will give us that indication if boating wouldn't be okay. We are hoping that we don't have to roll out the black tier.
Thank you.
Any other questions?
I would like to ask about your regulations for fertilizing lawns, which I consider a pretty big nutrient loader potentially, especially within, say, 100 yards of the water's edge.
So that might be a good question for Chris Murphy at the Conservation Commission.
I can answer it. It's easy. We don't have any regulation that involves homeowners. What we have is a regulation that involves paid people that are hired to come and take care of your property, put fertilizer on your lawn. They have to have a permit, a license. And the theory is, I guess you could take it away if they did it incorrectly. But they also have to have some level of education in the process. So right now, I'd say we're at a little above zero, but we're not at a high level of in any way regulating what people put on their lawns.
Amy, oh, go ahead, Marina, sorry.
Yes, if you're familiar with the regulations, they do limit the concentration, the season, how much you can use. And it's always been very difficult because even if we tried, we could not prevent stores from selling fertilizers that are more intense than we deem reasonable, especially for sensitive areas. We have tried to do public education around fertilizing for private, people who do their own fertilizing. We start with the landscaping companies because that's a lot of acreage and because we have the ability to enforce it, as Chris pointed out, which we do not have over the private citizen. But there it becomes a public education challenge.
Emily, might I speak once more?
Sure.
If it came out of this study, or all the studies together that are happening here, that was a major problem, then we can deal with it politically. Right now, there's no will to deal with it. But if you can say, we've got this problem, and the problem, and here's a smoking gun that points directly at what's being put on lawns, that changes everything. And I'm sure Marina and I and everybody else involved will go to bat for it.
I got to jump in here just because you're asking a question that drives me insane too. And I just wanted to inform this audience that the sampling and testing program in Chilmark Pond that's being sponsored by the Chilmark Pond Foundation is actually going to be able to identify the sources of nitrogen pollution in the pond. So the question, whether it's the geese or the swans or the imaginary cows or our septic systems, we should have that answer, or we should start to have that answer after a year's testing, right, Emily?
Right, we're partnering with the Marine Biological Laboratory in Woods Hole to look at nutrient loading, and one of the tests they'll be doing for us is running isotopes, so we understand the sources. And that'll be happening this year in Chilmark Pond, thanks to Chilmark Pond Foundation, Tisbury Great Pond, and Edgartown Great Pond. And Lori asked a question, is Is water that tests green still safe to the next round of testing a week plus later? I think this is a Marina question.
We go week by week because, as I was saying, it can change rapidly when the conditions are just right. So if you've got a green on your pond, go for it. The next week's sampling, if the next week's sampling changes the tier to a different risk level, I will be getting out there as quickly as I can to every access point I am familiar with, or that will give me access if it's private, to post the sign indicating what we have found that week. So when it's green, especially when it's green, you're in darn good shape. Remember, yellow does not say there is a bloom. Yellow just says that certain parameters are such that if, that a bloom is physically possible. It doesn't say that a bloom has been found. So if it's green, I'd say, the week is golden and keep an eye on it.
And after a season's worth of data, we're going to be able to see sort of trends. When we got to a certain temperature in this system, we tend to see this occur. This is how fast it changes. And we can start plotting the densities of blue-green algae, how fast they go, how the different things change. Having a season's worth of data, having a few years worth of data, we're going to be able to get more predictive with time, which is great. So for the data lovers out there, this is a wonderful thing. We also got a question about how this monitoring program is different than what's done by the Martha's Vineyard Commission. I don't want to speak about someone's testing program that I'm not doing, so I'm trying to see if Sherry is still here, Sherry Casso of the Commission. Sherry, are you still here? Julie, do you want to answer this question? Neither one of us are running the program, so we can only give you the basics that we've heard secondhand.
Sure. Yeah, I don't know all of the details, but I know it's similar to the Association to Preserve Cape Cod. The MVC is working with them and with some researchers at UNH to develop their program. They're not using fluorometry. They're more using visual inspection under a microscope to identify the types of cyanobacteria that are present. And if you see it's a species that is known to produce toxins, then they send those samples off to UNH for toxin analysis. And they're trying to develop what's called a regression to identify if there's a trend between the amount of a certain pigment in the water and the amount of microcystin toxin. So it's a collaboration with this one lab in UNH, and I don't know where exactly they're testing and how frequently they're testing. I believe it's weekly, but I don't know the details.
Okay. Are there any other questions? Wow, this is, oh, here we go, Kathy. No, you're saying bye? Okay.
No, I feel like I've been on a stiff learning curve myself, learning all of this, and certainly following the national news about cyanobacteria in the Great Lakes and along the coast of Florida. And so lots of people are paying attention, which I think is wonderful. But I also am really interested in looking for what are people doing, what to stop it. I mean, so yeah, yes, we know. So we're looking at nutrients and all of that. But are there other, are there things you can do proactively to keep the blooms from actually occurring? And I think I'm hoping that that's going to be a part of the picture that you're going to look at too.
That's the core of the work of Chilmark Pond Foundation, is figuring out not just for blue-green algae, which is important, but any type of harmful bloom. What drives blooms is heat and nutrients. We can't change the heat, But what you can do is you can reduce the nutrients. And Alan mentioned short and long-term solutions. So what we've seen work well at Evertown Great Pond is increasing circulation with pond flushes and increasing the effectiveness of each flush. What that does is in the short term reduces the nutrients, increases the circulation, increases the salinity, which also counteracts sort of the harmful species of blooming cyanobacteria. It can clarify the water and it can encourage other healthy things to grow. In the long term, you have to get into the watershed, and you probably do need to look at fertilizers. You need to look at septics and all the other sources, but having the data to know what percentage sort of the problem is coming from each source will let folks know what needs to be targeted first and how much. And so having those data to really match up both sides, the problem and the solution, will help. And what we've seen is a source of hope in Long Island because they have warmer and more eutrophic, more polluted systems. And with remediation, they've seen reductions in the intensity of algal blooms. That means reductions in the amount of cyanotoxins and the harm. And so remediation can work even in really polluted systems.
Do we have medical personnel discussing how the toxins affect human health? As in ingestion is one, but skin absorption or aerosolization and breathing, are these issues that we need to be concerned about? Or is there an authority that you have as a consultant on those issues?
Should I
think that's more you.
Yeah, okay. So my scientific knowledge is as good as anybody who is curious and stubborn and literate. I think there are different levels of human harm that are some of which are very well known. For example, getting when there is a toxic bloom, getting it into open skin, some of the neurological effects of long and intensive exposure or ingestion. These things are fairly well known. There's research being done on other forms of exposure, like intensive aerosolization if you're on the water, etc., that I don't think they've worked out. We are ourselves also fascinated by, but not yet clear on, exactly how it affects prey species of ours, not just fish and such, but animals that drink the water. Does it go for? Is it with the marine species? Is it only in the gut? Does it go into the muscle meat? When? How? Why? How quickly does it clear? These things we don't have an answer to. This particular Zoom is about this program. I would be delighted and excited, but it's very labor intensive to organize one of these Zooms. Maybe this would be a great off-season or early season Zoom to put together specifically about health effects, because there is a lot to be said about it, and very much I don't yet know.
And it's also an emerging field. There's lots of people trying to understand all the different types of cyanotoxins, what the toxicity is, and the scientists are still sort of duking it out about certain types of cyanotoxins. If you look at the scientific literature, there are new ones being discovered all the time, and it takes a multidisciplinary team really to answer questions about new toxins. So this is not an easy question, but it's definitely something that everyone's concerned with, and it's an emerging concern. One thing that might be helpful is we're hoping our colleagues at WHOI, I think Neil asked a question earlier, and Mark Hahn are trying to get a study funded about doing toxicity assays. Neil or Mark, are you still here? I see Neil.
Yes, I'm still here.
Neil, could you give us some context about toxicity and what your interests are?
Yeah, we are mainly interested in, we have been studying for a long time, looking at dinoflagellates, domoic acid, and saxitoxin, which are mainly from seawater have blooms. And I think we recently got interested in cyanotoxins because not just in Martha's Vineyard, Cape Cod, Falmouth, as well as other places on the Cape, the cyanobacterial blooms are increasing. So we are trying to use zebrafish as a model system, which is a biomedical species which is highly used to study what are the effects of exposure to chemicals. So I think we recently reached out to Emily and others trying to somehow test the waters and see, using a fish system, what are the effects of exposure, what kind of deformities we'll see, and then also understand some of the molecular mechanisms. So it is an emerging field and a lot of unknowns in terms of what are the long-term effects. We know that these are all neurotoxic, they affect the nervous system. However, we don't know much about what are the long-term consequences of exposure. Even the acute effects, we don't know clearly. So that's the focus. We recently wrote a grant, and if it is funded, potentially we can explore some of these questions.
And would you be looking at levels of toxicity that naturally occur in environments that you've measured, as well as higher levels that you've just simulated in the laboratory?
Yes. So trying to first, when we are trying to understand what are the potential mechanisms, we use a pure compound and do laboratory testing. At the same time, the things you guys are doing will help us know what are the environmental levels you see in different locations. So based on that information is really critical in order to do exposures in the lab at those levels, and then side by side compare what are the effects. And then earlier there was a question about what does the freshwater cyanotoxins do, whereas what about the brackish or the seawater cyanotoxins do? So it's the same question. There are people who have been studying cyanotoxins in freshwater for a long period of time, but we know nothing about brackish water and seawater cyanotoxins. So those are some of the questions which are interesting. And again, it's just unknown what the potential, how much toxin is being produced, how long it is produced, and are they same as freshwater toxins in terms of biochemistry? So there are a lot of unknowns.
We really hope that you're working on this project, Neil.
Yeah, no, we are really excited. Mark and I have been, you guys were amazing in terms of establishing a collaboration, and hopefully we'll have some samples and we can explore some of these questions. Thank you for organizing this. Yeah.
And Mark Hahn, I don't know if anyone knows him, is Neil's colleague, and he commutes on the ferry, and he's an island resident who is a scientist in the biology department at the Woods Hole Oceanographic Institution, as is Neil, but Neil's, I don't think, living on the island, so he doesn't get the same street credit.
That's true.
Does anyone? Oh, go ahead, Greg.
One more question. I'm interested in knowing what sort of information you're getting from your fluorometer. I take it you can detect cyanobacteria. Are you getting information that gives you an indication as to which species might be present? And can you extrapolate from there to say, well, this species is likely to produce X toxin, whatever? Can you get that sort of information?
So we have a microscope, so we can take images and send those images to colleagues who have sort of more experience. So there's two ways to tackle it. One, we have the scopes and the photographs that we can send off to people with that expertise, but also when we get to an orange or red tier, we're going to send our samples to Dr. Gobler's lab. And so he'll run the cyanotoxins analysis as well as look at some other things. So we're going to start working with collaborators to do some of those connections. That's not something we have the capability to do directly here. And also Dr. Fortunato will be preserving some samples for her in each of the systems sort of before, during, and after bloom season, where we can look at the genes to figure out who's present and in what abundance with some metagenomics. So it's going to take a bunch of different type of scientists to answer that kind of question. That one's not simple, but we're laying the groundwork to start trying to answer that. Did that answer your question, Greg?
It does indeed, but I take it that the fluorometer does not give you information about which species.
No, it does not.
Thanks.
It has a fluorescence specific to different types of plankton, but not species specific. Wouldn't it be great if it did? I think there's an $80,000 version that gets at that. Or at least they claim it does. Any other questions? Oh, Marina? Did you
Just
have-
a final note to say, I did not get the memo, but I so appreciate that you and Alan and Chris are all wearing blue-green t-shirts. And next time I will too.
That's right. Any future meetings, color coordinates. That's very funny.
Make the announcement.
I just want to say thank you, and it's a pleasure to be part of this community. Look forward to working with you all. If you've got bloom reports, send them to MV Cyano at greatpondfoundation.org. You can also go to greatpondfoundation.org and click on the report of bloom, and we will take that information and get it out to this whole network of scientists and health agents, because we are all meeting weekly and we're committed to getting the data out as quickly and effectively as possible. So thank you all, and happy summer.
Thank you, Emily.
Sure.